Okay, everybody, so we're going to set up and do the growth curve right now. And the only difference with the growth curve from the enumeration is going to be that you're going to do one extra step. So what I've done so far is I have made my dilution series, just like we did for enumeration. And then I got to set up the spectrophotometer. I got my plates ready for doing my hockey sticking, and then my hockey stick is right here, etc. I got my tubes.
Don't! Those are tips!
Okay, everybody, so we're going to set up and do the growth curve right now. And the only difference with the growth curve from the enumeration is going to be that you're going to do one extra step. So what I've done so far is I have made my dilution series, just like we did for enumeration. And then I got to set up the spectrophotometer. I got my plates ready for doing my hockey sticking, and then my hockey stick is right here, etc. I got my tubes. I've got a cuvette. And so what the point of this is going to be, you all will get a culture. It will be on the shaker. And you will basically, at each time point, we're going to shoot for probably time 0, 15 minutes, 30 minutes, 45, and an hour. We're going to shoot for that for each of you. And we might modify those times a little bit as we go to see how things grow. But what you're going to do is they're going to be growing on a shaker or in a different environment in a flask. And they're going to be at a predetermined concentration initially. So at time 0 and at each time point after that, you get to keep track. You have to know the time between time points. Like it's every 15 minutes, set a timer. So when you decide to do time 0, you're going to go over to the incubator shaker and you're going to grab your flask. And you're going to bring it back to your bench and be as efficient as possible because you're taking a time point, meaning once you take that flask out of the incubator shaker, it's now out of the environment where they're growing. So you have to make sure you do the rest of this pretty efficiently so you keep moving along. So for example, when I grab my flask, I'll bring it here and then I will not do all of these manipulations while leaving the flask out. I'll take the flask, I'll take my samples out, I will put flask back on the incubator and let it keep going because you want to keep that on the incubator and shaker as much as possible, okay? Or whatever your particular condition is going to be or be assigned. All right, so what we'll do first is blank the machine. So in my case, I'm growing in something called CTT medium, which is this stuff here in the flask. So I, of course, had to use this to blank my machine. To blank this thing, you take your cuvette, and again, you don't want to touch the bottom part of this cuvette unless you're touching the very edges. You want to keep the other parts wide open and clean from dirt and oil. If you're nervous, go ahead and use a Kim wipe or something and wipe it off. Some of these have a little arrow on them that shows you which way to point it. Now you come over to the little spectrophotometer. I would turn this thing on initially and let it warm up for a few seconds before you start doing anything. On button is right here on the green. And in this case, if you don't know where the light's coming through, it could come this way or it could come this way. So you got to put your cuvette in the right way. Just hit the R button for the blank or the read button, and you'll look down there and you'll see a little light come on. See that little light? So that means our light is coming this way. So that means we want to make sure that you put the path of this thing so that it's clear and that these flat pieces are going direction of the light. So I've put in my I put in 900 microliters about, so I make sure you have to have your liquid high enough so that it goes over that little light. Okay, if it's too low, it won't read. So I know I'm okay. I'll stick this in here like so, down to the bottom until it snaps, and then I'm going to hit the the blank button, and you'll see this little thing go to 0. So there's my blank is done. Now I'm ready to go. Once this thing is blanked, it's blanked for the duration. You shouldn't need to reblank it. Now, you're either going to use a new cuvette or you can take this and dump it in the biohazard bag. In my case, I'm going to use a new one. because it's just simpler in this case. What we're going to do now is go get our sample and do it just like you would do it in your experiment. So it's time 0 right now. Bam. So I have to go get my sample. Here we go. Waltz over here to the shaker and lift this up. Grab my sample. Keep it shaking. Now, fairly quickly here, you want to take samples out of this, and you should use sterile technique to do this because you don't want to mess anything up. I gotta go turn the gas on. Can't light the fire. There we go. All right. So, I am going to use sterile technique and remove this sample. I only have to remove about 500 microliters. If you're nervous, go with 600, but you don't need a lot to put in this cuvette. Okay? So, take your appropriate tip, use sterile technique, carefully grab your sample and put it in the cuvette. I'm also at the same time going to take 100 microliters out of the sample because we're going to do our 1 to 10 dilutions. Sterile technique. Put it in my labeled and ready to go little dude. Now, before I do anything else, I'm going to go ahead and put this flask back because it's now not shaking. I want to put it back on the sample, on the incubator. I took my 2 samples, my 600 microliters or 500 microliters for the cuvette, and my 100 microliters for my first dilution. So put this back as soon as possible. In fact, you should put it back a lot quicker than I'm doing this. This thing will turn off on its own when you lift it. Put it right back in. Boom. Now you're ready to do everything else. Okay? In this case, the most important different thing compared to what you did before was take your little cuvette, make sure it's the right direction, put it into your blanked spec, and hit the read button. And we get 0.51 in this case. So I'll put that down in my lab book. And so I know that at time 0, I'm at 0.51. 15 minutes later, that's already going, I'm going to do the very same thing. Okay? Now I'm not going to show you this, but now you immediately make a serial dilution of this, and you plate it on your plates with the hockey stick, just like you did before. Have these all pre-labeled. And now for each time point, you're going to have CFU data in a day or two, and you'll have instantaneous spec data that you'll be able to graph immediately. Okay, so you're going to hopefully get, say, 4 time points in the hour and a half, and you hopefully will see a difference in this optical density, and that will show growth or no growth, depending on what's going on. Okay, so that's as simple as it is. Next thing we'll do is show you how to graph the data once you get it.